This would be this is the length. So this muscle cell, this is one muscle cell, actually more than one cell because it's right on the edge. Here's one cell and here's another cell with the nuclei just under the plasma membrane. And so this is actually a whole bunch of myofibrils. And every myofibril lines up. So its A band is in line with the A band of the myofibril next to it. And so when it shortens, remember these are getting narrower, so the muscle cell shortens this direction. So if we look at, so this is a myofibril, all right, but in a muscle cell, this myofibril Z disc is going to line up with this myofibril Z disc and so on, so the entire muscle cell shows that pattern. Yes?
During contraction, do the actin ever touch each other or touch the...
They can't actually overlap each other. So you can eliminate. So we can see that in this image. Here. So there's actually the actin, which is the blue line here, is overlapping. And it's eliminated the H zone. And practically eliminated the I. It's about eliminated the I band as well. All right, so here is a muscle cell at rest. And you can tell that this is an image that's contracted because it's shorter. So the two Z lines are closer to each other than they are here. So that's obvious. But if you look at it, here the H zone is narrow. And the I band, you're only seeing part of the I band here, but that also, the distance from the Z line to here is narrow. So that's going to tell you, this is an electron micrograph, but you can see almost as much detail with a good light microscope. Now on the lab activity, to make you focus on this, I actually used the ability to adjust pictures. And I actually stretched out the sarcomeres, so both sarcomeres are the same length. So you'll have to focus on the H zone and the I band to determine if the muscle cell is contracted. This is too obvious that it's contracted. So I got to stretch it out so the sarcomere is the same length, and that's not going to be the case. But I wanted you to focus on changes here and here. Now the diagram at the bottom of the page, or the bottom of the screen here, is showing the efficiency of contraction. So obviously if we start out our contraction with the muscle cell so relaxed that the actin is barely over or not even over the myosin, the heads pulling on that actin filament are not going to be able to create much tension. And if you start with them almost touching, they're not going to be able to create much tension either. So there's a maximum separation where there's enough filaments with the heads available to grab the actin and pull to create the maximum force or tension. And that's what's being illustrated by the graph. There's an extreme in either condition, where they're so far apart or too close together. Next semester, when we talk about the heart, we'll talk about a membrane that forms a sac around the heart. Helps to lubricate the movement of the heart, but it also keeps the heart from filling up too much with blood. Because if the ventricle fills up too much with blood and stretches, guess what it's going to do to the actin and myosin? It's going to separate the actin and myosin and pull them apart from each other so far that myosin won't be able to grab a hold of it, or if it does, can't create enough tension to contract and move blood out of the heart efficiently. So when that day comes, I'll remind you of this. Maybe it'll be warm outside. Actually, it'll be February when we talk about the heart. Okay, so let's look at how a muscle contraction occurs. So that was kind of anatomy. And half of this physiology you already know. It's nerve physiology, even though we're talking about muscle action potential. You already know how it happens. which is what I find interesting that in every anatomy and physiology textbook, they talk about muscle physiology first before they've laid down the groundwork of what resting membrane potentials even are. So they ask you to understand an action potential before you even know how resting potentials are created, which is why I switch, one of the reasons why I switch all of that. So can I erase the board? May I erase the board? Not quite yet? Okay. Thank you. Uh-huh. All right, so anybody remember the term that we use for the relationship between two neurons? One neuron activating the second neuron? What do we call that relationship between the synaptic bouton of one neuron and the dendrite of the second neuron? What has been formed? A synapse, all right? We have the presynaptic membrane and the postsynaptic membrane. Well, when we talk about a nerve and a muscle cell, a different term is used, even though the relationship is very similar. And this term is a neuromuscular junction. A muscle cell, like a nerve cell, is irritable, meaning it can respond to a trigger by creating an electrical impulse, and it has a conductive membrane. We have voltage-gated channels. In addition, the muscle cell can contract. All right? Nerve cells don't have the myofibrils. They can't contract. They're irritable and conductive, but they can't contract. So we have our neuron, and this would be a lower motor neuron. Coming its cell body is going to be in the anterior gray horn. And one of the axon terminals expands as the synaptic bouton or end bulb. And stored within that, we have synaptic vesicles, which can fuse with the presynaptic membrane. It is going to interact with the sarcolemma of the muscle cell. This is specialized because it has ligand-gated channels instead of voltage-gated channels. So this specialized membrane is known as a motor endplate. It is the sarcolemma with ligand-gated channels. It does. I just haven't gotten to that point yet. Okay. So this would be equivalent to what region of a neuron? What area of a neuron has ligand-gated channels? The dendrites and the cell bodies. Okay? Sorry, it's getting hot in here again. It doesn't get too noisy in the other room. So, usually, one neuron, remember how it branches out and has multiple axon terminals, will innervate more than one nerve. Fine motor control, it'll only activate maybe 3, 5, 7, 8 nerve muscle cells. Fingertips, lips, where you can do fine muscle movement. Muscles in the back, one neuron will probably activate several hundred muscle cells. All right, so we call that a motor unit. So it sounds similar to motor endplate, but a motor unit is equal to a single nerve cell plus all the muscle cells it innervates. And the lower the ratio, the better the muscle control, the fine motor control. Okay? So I just wanted to differentiate the motor unit versus the motor end plate. So we look at this a little bit closer. And so we still have a synaptic cleft here and synaptic gutters, these folds. And so the more folds we have, obviously, the more surface area there is and the more ligand-gated channels we have. Right? So you decide that you're going to move a muscle. I simulated somebody this morning on my way to work to move a muscle. He gave me a finger.
Mm-hmm.
He cut in front of me, and I blew my horn at him. He cut in front of me from 3 lanes over. And, yeah, so I blew my horn at him, and he slowed way down. And I was in the carpool lane. I was like, fine. You want to go slow? I can go slow. You're the one that moved in the fast lane. So I slowed down and turned my brights on. My son gets upset with me when I do that. And so I was like, if you want to go 60 miles an hour in the carpool lane, that's fine. I've got plenty of time. You're the one that pulled into the lane. And so then he sped up and proceeded for the next, until he got off at the exit, to alternately, I don't know what he was trying to do. He was braking, but he was like 300 yards in front of me, so just brake on and off. And then he started to turn his lights on. So he turned his left light on, his blinker, right light, left light, right light, left light. I was like, okay, I don't know what that means. Anyway, I triggered him to make a conscious movement and bend his finger and extend another finger, all right? So his lower motor neuron was activated, and an action potential came down the axilla and the other way. Came down the axilla and down the axon terminal and got to the synaptic bouton. And when it arrived at the synaptic bouton, what kind of channels were open?
Calcium.
Calcium. Voltage gated or ligand gated?
Voltage.
Voltage gated. So calcium, voltage gated channels. And that allows calcium ions to diffuse into the cell. And as a result, anybody remember the name of the molecule that binds calcium in the synaptic vesicle and allows it to fuse? Starts with an S, has the word tag in it. Synaptotagmin. So calcium binds to synaptotagmin. It's part of our synaptic vesicle. And that allows the synaptic vesicle to fuse and release the neurotransmitter. What's the neurotransmitter in our lower motor neuron? It's the same neurotransmitter that we see in the parasympathetic, preganglionic, and postganglionic neurons. What is that? Acetylcholine. Okay? So acetylcholine is released across the synaptic cleft in response to the calcium ions moving in. And our motor end plate is specialized because it has ligand-gated channels. Are these ionotropic or metabotropic? Is this a nicotinic or a muscarinic receptor? It's excitatory. It's very quick acting to open and close. Quick acting, quick closing. Quick opening, quick closing. Ionotropic. So nicotinic. All right, so this is a nicotinic. Remember, acetylcholine has nicotinic or muscarinic receptors. Where else do we find nicotinic receptors? Postganglionic, sympathetic, and parasympathetic fibers. Okay, so in the ganglia, whether it's a ciliary ganglia or a paravertebral ganglia, the preganglionic cell, either it's sympathetic or parasympathetic, releases acetylcholine. And so the receptor for that on the postganglionic neuron, whether it's parasympathetic or sympathetic, is a nicotinic acetylcholine receptor. That's why cholinergic, too much acetylcholine can affect both sympathetic and parasympathetic and cause convulsions, because those are all nicotinic components. All right, so this is a nicotinic receptor, and by definition, then all nicotinic receptors are ionotropic. They create excitatory postsynaptic potentials, allowing sodium ions to enter the cell. So acetylcholine binds to its receptor, and sodium ions enter the cell. This creates a potential. Is it an action potential or a graded potential? When we open a ligand-gated channel, it is always,
Action.
exactly right question, a graded potential. Voltage-gated channels are action potentials, okay? But ligand-gated channels are always going to create a graded potential because it depends on the amount of neurotransmitter that releases. Small amount is released, a small potential occurs. A large amount is released, a large potential occurs. Okay? So these ions will travel this direction and this direction to reach the rest of the sarcolemma. And now this sarcolemma has colors here. Has voltage-gated channels. So the rest of our membrane is going to demonstrate voltage-gated channels for sodium and potassium. So are we going to have a muscle contraction with every activation of our lower motor neuron? Anytime our lower motor neuron releases acetylcholine, is our muscle cell going to contract? No, because it's a graded potential. Okay? So I can go move, 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 and it doesn't move. Because I didn't release, I don't know how much, I can't tell. But I didn't release enough acetylcholine. Okay? How many of you did on the PhysioX did 8.0 instead of 9.0? All right, so you guys used the curare, the experiment with the curare. Curare is a neurotoxin, well, it's a toxin, it's not really a neurotoxin, that is derived, it's made famous, it's used by other countries. Natives in Borneo use it, but it was made famous by native South Americans that would paint the tips of their arrows with it and go after food, game, and when they shot the game, it wouldn't kill them, but it would paralyze them, and they would fall down, and then the hunters could go and actually kill them by some means, club them over the head or whatever. So if you recall, and it was a little while ago, when you used the curare in the experiment on PhysioX, did it prevent the neuron from creating an action potential? It did not. The reason is it doesn't affect the nerve. Curare blocks the muscle like acetylcholine receptors. So the nerve is still firing and acetylcholine is being released, but with curare it's blocking the channel on the skeletal muscle. Okay? So it's not interfering, that's why I said it's not a neurotoxin. It's not interfering with the nerve activity itself. All right, so let's say now we've created our graded potential. It's sufficient that when that graded potential reaches the voltage-gated channel, it reaches threshold, and it opens and starts an electrical current, as the action potentials are self-generated. Now, this sarcolemma extends deep into the cell, so guess where that current goes? Deep into the cell. So this is our transverse tubule. And remember, the transverse tubule contains extracellular fluid. So when the voltage-gated channels open, they allow sodium from the extracellular fluid to flow in as if it was on the surface. So our action potential travels along the plasma membrane into the cell. That was the whole purpose of having it do so. And on, what's that supposed to be? I swear, all of these things have the wrong cap. So right next to the transverse tubule, we have the dilated regions of the sarcoplasmic reticulum known as the terminal cisternae. And what was unique about the plasma membrane of the terminal cisternae? Remember, I related it to the axon hillock? And what's unique about the axon hillock? How did the axon hillock differ compared to the axon itself, as far as the voltage-gated channels and their properties? It was easier to trigger the action potential because the threshold was lower. It took less of a current to open the voltage-gated channels at the axon hillock. Same thing for the voltage-gated channels in the terminal cisternae. It takes less current, and it gets that current from the transverse tubule. So the current that's entering into the cell from the transverse tubule is right next to the sarcoplasmic reticulum. And not only will it open channels down the sarcolemma, it will open channels in the sarcoplasmic reticulum. Once those open, it spreads along the rest of the sarcoplasmic reticulum. And these are voltage-gated channels for calcium ions. Is calcium going to go into the sarcoplasmic reticulum or out of the sarcoplasmic reticulum when those channels open? Out. So we had calcium channels up here in the synaptic gluton. And the calcium bound to the synaptotagmin and allowed the vesicle to fuse and release acetylcholine. That calcium came from extracellular fluid. Okay? The current moves deep into the cell, activates voltage-gated calcium channels in the sarcoplasmic reticulum, and that's going to allow calcium ions that are stored in the sarcoplasmic reticulum to move into the intracellular cytoplasm. Not outside the cell, but outside the sarcoplasmic reticulum into the intracellular cytoplasm. So levels of calcium, free calcium in the intracellular cytoplasm will be elevated. They're no longer, or fewer, are being stored in the sarcoplasmic reticulum. So what we've seen so far is this activity here. So calcium ions are being released. All right, I'm going to erase this half of the board, so if you want to take images, yes.